This is the recording for Chapter 2, Part D. So it's the last section of Chapter 2. It'll also have some material from Chapter 26 because that has part of the metabolism material. So our third group of organic compounds are proteins. In this group, you'll have two different structural formulas to identify. Now, proteins is both the name of the group and it's the name of the polymer. So proteins are organic compounds. They have carbon, hydrogen, oxygen, and nitrogen. Some have phosphorus, sulfur, and iodine. They have the two functional groups that are always present, amino and carboxyl. We also define proteins as chains of amino acids. They're a type of polymer. They have a special type of covalent bond called peptide bond. So with proteins, you'll see dipeptides, tripeptides, oligopeptides, and polypeptides, each with 1, 2, 3, or more amino acids in a chain. The shape and sequence of amino acids determines the ability of the molecule to function. If it doesn't have the right shape, it cannot do the right, it cannot work. It cannot carry out a function. Now, here you have a simple representation of amino acids in the form of balls. Oops. Sorry. Oops. So these simple balls represent a single amino acid. And so when we make chains of them, down here you get proteins. This shows an extended version where the chain starts curling together, or it can fold back and forth. Again, with proteins, they have to have a specific shape in order to carry out their function. So the amino acid is the monomer. You have 4 examples of amino acids. Amino acids are composed of an amino group, a carboxyl group, and what we call the R group, which includes this carbon and hydrogen. In pink, they're highlighting the differences in the R group. The capital R stands for the radical portion of the molecule. There are 20 different amino acids. Here are 4 of them. The simplest R group would be an H with the carbon and another H. But these you can see vary. They can have a carbon ring like we see, down here. They can have a carbon chain with nitrogens. So they can vary. So you need to be able to recognize any of those as an amino acid. The peptide bond is a special covalent bond between the carboxyl group of one amino acid and the amino group of another amino acid. So down here, highlighted in green, we see a peptide bond. So it has one of each of the four most common elements in the body. Again, we have to take off water in order to put the molecule together. So you'll see dipeptide, tripeptide, polypeptide. And what you're going to label, it would just simply be a protein. So we're just doing amino acid and protein. Technically, it's a dipeptide, but you don't have to get closer than that. Okay, so because structure is so important, we have 4 different levels of structure. So you've got a table to fill in. The first level of structure is called the primary structure. The primary protein structure has a single chain. So number of chains is 1. The type of bonds is peptide bonds. For the shape, it's a long, narrow strand. And no, it does not function. This is what comes out of the ribosome. Next, you have secondary structure. That would be the next level. It also has a single chain, so one chain of amino acids. Type of bonds is peptide and hydrogen bonds. So, second level of structure, 2 kinds of bonds. There are 2 shapes. There is what we call the alpha helix, which is this coil. Oops. And the beta sheet, which can look like this or like it shows down here. Oop. There we go. The folded sheet. So it can fold back over on itself. The little red dotted lines represent the hydrogen bonds. And no, they cannot function. So as they take up, as the chain increases in length, it can only take up so much space. And so it starts coiling up or folding back and forth. Next is called tertiary. It also has a single chain of amino acids. It has peptide and hydrogen bonds And then it also has ionic bonds, but only between R groups. Two kinds of shapes, a complex globule, like we see here, this cloud-looking thing, or you can find them in long filaments. Yes, these function. The cloud is something that we would see like with, or complex globular would be like an antibody, an enzyme. So this is when they start to function, when they reach tertiary shape. The fourth level is called quaternary structure. There are two or more separate chains of amino acids. Shapes, bonds is the same as tertiary. So there are peptide bonds, there are hydrogen bonds, and ionic bonds between R groups. Again, they can either have a complex globular shape, like we see here with hemoglobin. There are, or the filamentous shape, which is what we see over here with collagen. In hemoglobin, you have, one, two, three, four amino acid chains, and you have four hemes, which are the four red flower looking things. Those are pigments. So this is a complex quaternary protein. Yes, it functions. Collagen, they used to think it was a tertiary protein, but they got better imaging, and there are three different strands. So you've got the dark brown one, and then two light colored, and they're twisted together like a rope. So the only difference between tertiary and quaternary is the number of strands. You have the same kind of bonds, same kind of shapes. Yes, they both function. Currently, there are just the two examples of quaternary proteins. It used to just be one. Again, because shape is so important, you have the term denaturation. Loss of shape in a protein because of high temperature or a change in pH levels. Sometimes the change is permanent, and in others, it's temporary. An example of temperature, when you cook an egg, the clear, slimy part of the egg, when you cook it, becomes white, opaque, semi-solid. When it cools off, it doesn't go back to being slimy and clear. That would be permanent denaturation of the protein albumin. With pH, it can also be permanent or temporary. In the mouth, oops, here we go. In the mouth, you have amylase. That's a Y. Why this? I don't know why it's so lit. Anyway, Let's pause. Okay, so in the mouth, you have amylase. This is a protein that digests starch if it's the right pH. In the mouth, you have a high pH. And so it works. Once this material goes down into the stomach where the pH is low, then the amylase is denatured permanently. There's another enzyme secreted in the saliva that's called lingual lipase. It doesn't work in the high pH of the stomach. When it reaches the stomach, however, in the low pH, it does work. So it begins to work, breaking down lipids. As they enter the small intestine, the pH is going to slowly increase. And so then it becomes denatured. So with lipase, it's a temporary condition, depending on the pH. High temperatures, when you have a fever, the problem with them is if they get too high, they can denature the myelin sheath in the nerves, and that causes permanent nerve damage. A lot of times, if children have had too high of a fever as a child, they'll start developing seizures the next time they have a fever. So it's a side effect because of the high temperature and damage. Okay, so those are our three organic compounds: carbohydrates, lipids, and proteins. We're also going to take a look at another group, at least part of another group. The nucleic acids have a monomer called a nucleotide. One commonly found and discussed nucleotide are ATP. The ATP is called adenosine triphosphate, but yes, you can use that abbreviation because it's in the list on your syllabus. It is composed of adenine, which is this orangish beige color, ribose, which is the blue, and three phosphates, which are in the purple here. You have 1, 2, 3 phosphate groups. It's called a nucleotide because it's got those components. It could be used to form a nucleic acid, like DNA or RNA. This particular nucleotide is used by cells as a form of energy. By removing one phosphate group, you can release a larger than usual amount of energy. If we look closely in the chain of phosphates, right? Oh, shoot. So what do? Erase it or cover it up. If you look closely, right here, you have a squiggly line instead of a straight line for the bond. Same thing here. These are higher energy bonds than we typically see. So if you break off one phosphate, you release the higher than usual amount of energy. If you break off a second one, you get also a higher than usual amount of energy. So that's why ATP is considered our energy molecule or our energy currency. Cellular respiration is how we make ATP. There's a generic or equation, and there's also some specific equations. Cellular respiration is defined as conversion of organic compounds into ATP molecules. You can use various different compounds. Glucose gets you the most ATP. We can use other monosaccharides, fatty acids, glycerol, and amino acids. But again, you get fewer ATP molecules if you use them. This is an overview of cellular respiration of glucose and the products. Oops. Cellular respiration has an overall or generic chemical formula. Yes, this is the one you have to memorize. And yes, you have to make sure there's subscripts. C6H12O6 is glucose. Plus 6 molecules of oxygen. It's in parentheses, so you don't think that it's 6, 0. You have an arrow going across, producing 6 molecules of carbon dioxide, 6 molecules of water, and 26 to 38 ATP. Now, the number of ATP is going to depend on the cell and how efficient the mitochondria are, because that's where cellular respiration is going to take place. In bone cells, which are not as active as a lot of other ones, they're going to be in that 26 range. Muscle and liver cells, which do a lot of activity, are going to be in the 38 range. So it depends on the individual type of cell and how active they are, how frequently they carry out cellular respiration. This is another video that you need to put on your list, the biochemical pathway. It shows you how chemical reactions take place in a generic form. Also, how glycolysis works is another video that you need to watch. It shows you the first of the reactions. Oh, I think I missed an objective back here. The ATP cycle. Let's see if I can get rid of some of this. Okay, the ATP cycle is, oh shoot, ATP P A D as in dog, P plus an inorganic phosphate. So it's a cycle. You're going to do a curved arrow and another curved arrow. Now, to finish this off, ATP with an enzyme will break off one phosphate group. When we do that, we release energy. And that's the energy okay, released. And then we can use it. Okay, so then when we put phosphate back onto ADP to form ATP again, it's going to require energy. Now, of the energy that's released, so this stuff over here, typically we can use somewhere between 10 to 30% is usable. The other 90 to 70% becomes heat. And that goes back to physics and chemistry, the laws of what we call thermodynamics. Part of it's always going to become heat, and in our case, most of the time. Now, 10 to 30% usable energy may not seem like a lot, but if we compare our cells to our car or truck or SUV, whatever it is that you drive, they are only 2 to 3% usable energy, and the rest becomes heat, which is why you have to have a big radiator to dissipate the heat. Now, we use that heat to maintain our body temperature. So, not all bad, but technically heat is a waste product. Okay, so let's get back to the rest of cellular respiration. Okay, so there's your overall chemical formula. Two pathways or two videos to look at. Here's an overview, and it compares where it takes place in the cytoplasm or cytosol and the mitochondria. It breaks it down into different steps: glycolysis, the transition reaction, citric acid or Krebs cycle, and the electron transport chain. So again, you have a table that we'll fill in. Here, however, it shows all of the individual reactions included in glycolysis. At the end of glycolysis, it depends on whether or not you have oxygen as to what's going to happen next. You can go into anaerobic fermentation, or you can continue to aerobic respiration. And yes, this part is a duplication of this, but you needed to see the transition. So the farther you go into biology, the more of this pathway you have to learn. So trust me, you're getting off easy. This shows the transition reaction over here in this blue box, and green the Krebs cycle, which is also called the citric acid cycle, and also called the matrix reactions. And then down here is the electron transport chain, which is also called the membrane reactions. Okay, so you have a table. You need to put the following numbers to the left of the table so that you know how many times the reactions occur per glucose. Glycolysis happens once. Transition reaction happens twice. Krebs or citric acid cycle happen twice. The electron transport chain is going to happen 12 times. Okay. For the first row, glycolysis is the name of the reaction. It happens in the cytoplasm. The principal reactants, and yes, you have to know all of them. Glucose, 2-ATP, 4-ADP, 4 inorganic phosphate, 2-NAD+. Principal products. You'll note that there's some columns, and that there's also a other products list. So under products, pyruvic acid, there's 2 of them. That goes under other. 2-ADP, 2 inorganic phosphate, and then you'll put the number 2 under NADH, 2 water, and 4 ATP. Note over to the right, there's only a net gain of 2 ATP because we start with 2 and use them, and we generate 4. So you're only getting 2 new ones because you used 2 to start with. Okay. Second part, subreaction, which happens twice per glucose, called the transition reaction. Principal reactants, 2 pyruvic acids and 2 NAD+. Notice that 2 of the pyruvic acids were made by the previous reaction. Products, 2-acetyl-CoA. So you just need to put the number 2, 2 in next to the acetyl-CoA. And that's short for acetyl coenzyme A. And then 2 in the NADH column. So this is the total numbers, but the reaction actually happens twice. So each pyruvic acid is going to be converted into a coenzyme, to acetyl-CoA. Third, Krebs cycle or citric acid cycle. Reactants, 2-acetyl-CoA, put the number in front of that. 6 NAD plus, 2 FAD, 2 ADP, and 2 inorganic phosphate. These will all go in the columns. So 6 NADH, 2 FADH2s, 6 carbon dioxide, 2 ATP. Again, these are the total numbers. The last reaction is the electron transport chain. It's also called the respiratory chain, the cytochrome system, are the three main ones. Principal reactants, 6 oxygen molecules, 10 NADH, and 2 FADH2. So this is going to happen 12 times, one for each of the NADH and one for each of the FADH2s. Products, 6 water, and somewhere between 22 and 32 ATP. Again, it depends on how efficient it is as with regard to the cell and how often the mitochondria is used. Okay, now we're going to go back and look at this with regard to our general equation, because you need to know where that comes in as well. So here in glycolysis, we see glucose. We see the production of 2 new ATP and the products. Transition reaction doesn't contribute to the general. In the Krebs cycle, you get the 6 carbon dioxide and 2 more ATP. In reactants, this is where the oxygen is used, and this is where the water is produced. Yes, they put markers on the atoms and trace them. And then this is where most of the ATP molecules occur. All right. Then you have the terms anaerobic and aerobic. Anaerobic without oxygen, aerobic with oxygen. So fermentation is cellular respiration that occurs when you don't have much oxygen. You produce fewer ATP. You produce more lactic acid if you're animals. If you're plants, you would make alcohol. So, difference between animals and plants, the waste product produced. Now, lactic acid is what we consider a waste product. It's also associated with the burning sensations that you have in muscles when you overuse them. Aerobic respiration, cellular respiration that occurs when you have lots of oxygen, you produce more ATP, and you produce less lactic acid. So that's why a lot of times with muscle activities, they include when to breathe, or including reinforcing the fact that you need to breathe. All right, so that marks the end of the chemistry chapter.